video module, we are going to learn about an extremely interesting bacterium, namely Nocardia, which I personally call as the cards player. Now, first and foremost, the organism, the bacterium Nocardia, has been named so after a French veterinarian, namely Edmond Nocard, who described this organism for the very first time in the year 1888. Okay. And what kind of a bacterium is this? Remember guys, Nocardia jo hai, it is a type of a gram-positive filamentous bacterium. Gram-positive filamentous bacterium. That is just like the filamentous fungi. This bacterium also morphologically exists as, filaments or hyphae, and that is precisely why this organism was initially it was confused to be a fungus, but later on it was discovered that this fellow has got what? Bacterial, that is prokaryotic ribosomes. Okay? It was found to have prokaryotic ribosomes, and it was found to respond to antibacterial agents, and it was concluded that this fellow is a bacterium and not a fungus. And we have got another important gram-positive filamentous bacterium which you need to know for your exams, and the name of that fellow is Actinomyces. Actinomyces. There are 5 important genera which come under the category of gram-positive filamentous bacteria, and amongst these 5 genera, the 2 most important for your exams which are asked in detail are Nocardia, which we are going to study in this video module, and Actinomyces, which you can check out in a separate video module of mine. Okay? Now, so because these 2 are the most important genera of gram-positive filamentous bacteria which you need to know, that is Nocardia and Actinomyces, it's very important to learn the differences between these 2. So let's learn the differences quickly. Oxygen needs. Remember, Nocardia is an aerobic fellow, it's an aerobic organism, whereas Actinomyces, those of you who have the habit of reciting out loudly when you are reading your notes, they will relate to this. When we say actino, जब हम actino का उच्चारण करते हैं, it sounds very similar to what? Anaero. So actino, anaero. So Actinomyces is anaerobic. This is one very important difference between these 2 bacteria. Acid fastness. Nocardia is partially acid fast. Partially AF, that is acid fast, and 1% H2SO4 is used for Nocardia during modified Zn staining if you are suspecting Nocardia in any clinical specimen, whereas Actinomyces is non-acid fast. NAF means non-acid fast. Okay? Then disease in whom? Remember, Nocardia causes disease in I/C, that is immunocompromised individuals usually, whereas Actinomyces causes disease in immunocompetent individuals. Okay? Another important point of difference. Then infections. Infections of Nocardia are exogenous, meaning the infection is acquired from outside. Remember, Nocardia jo hai na, it is never a commensal, it is present as a saprophyte in the environment. More on that in just a bit. So when the organism Nocardia itself is not present in the human body, it is not a commensal, so it is acquired from the outside, that is environment, and that is why we say that the infections are exogenous. Bahar se acquire kiya jata hai ye organism. Then what about Actinomyces? Remember, most of the infections of Actinomyces are endogenous in origin because Actinomyces is found as commensal flora in the human body. ठीक है, तो commensal की तरह exist करता है नहीं करता है? Yes, it does exist as commensals. For instance, in the oral cavity, in the gastrointestinal tract, etc. All of this I have covered in the video module on Actinomyces. So please keep in mind these important differences between these 2 most important genera of gram-positive filamentous bacteria, Nocardia and Actinomyces, which you must know for your exams. Have a look at this, guys. Nocardia. Like I said, Nocardia is an example of a gram-positive filamentous bacterium. It exists as, gram-positive filaments. So it was initially confused to be fungi. So its appearance under the microscope is a little misleading, and as we all know, appearances can sometimes be deceptive or misleading. So Nocardia is telling you this, whatever this schematic diagram which I have drawn is representative of Nocardia, these blue filaments. Nocardia is telling all of you ki I am not what I look like, just like certain people are. Okay? So on gram stain, Nocardia appears as gram-positive filamentous, bacteria which are 0.5 to 1 micron in width. Okay? An important, difference between the hyphae of or the filaments of Nocardia and fungi is that the filaments of Nocardia are pretty narrow. Remember, the width of the filaments is, less than 1 microns, and fungal filaments usually are broader than this. If we talk about the residence of Nocardia, remember it's an environmental saprophyte found in the soil and vegetations. Okay? And the species which you need to remember in the genus Nocardia, 3 most important species include, remember I have written them as A, B, C, the first alphabet of the species, namely Nocardia. A से है asteroids. B stands for Brasiliensis. C stands for KVA. Now asteroids को asteroids क्यों बोलते हैं? Because of the star shaped colonies it produces. ठीक है? Star shaped colonies. Brasiliensis शब्द का literal meaning is pertaining to or related to or found in Brazil. KVA pertains to what? It pertains to guinea pigs, because this very species Nocardia KVA was initially isolated from guinea pigs. Have a look at this, guys. So at the outset of this video module, I mentioned that Nocardia is the cards player. ये मैंने खुद का नाम दिया इसको. तो let's see how does it play its cards. मतलब ये humans को कैसे infect करता है. I have highlighted N and C in the name Nocardia. From N and C, please remember nose and cutis, मतलब skin, pertaining to skin. ठीक है? So it likes to play its cards via nose and cutis. Remember, this fellow Nocardia, as I've drawn in blue color over here, it's a gram-positive filamentous bacterium. It's a saprophyte in soil and vegetation. So it is, exposed to the environment. So just remember, this is a little memorizing tip, that these fellows are obligate aerobes. So ये environment में exposed हैं, present हैं, तो oxygen इनका कुछ बिगाड़ नहीं पाती है. They in fact love oxygen. They are obligate aerobes. They need oxygen. So this is one important point of difference between Nocardia and Actinomyces also, which I said earlier. ठीक है? So nose and cutis मैंने यहाँ पे क्यों mention किया है? Because these saprophytes which are there in the soil and vegetation, how they infect human beings? So let's write quickly the modes of infection. The modes of infection include, number 1, inhalation of the fragmented bacterial filaments. Okay? So via nose, the bacterial filaments which are there in the environment, they are entering the human body. And the second mode of infection is the transcutaneous inoculation, okay, which leads to various cutaneous and subcutaneous manifestations. Okay? So let's say some trauma happens with something, and that something, some environmental object, some vegetation and all, they are harboring Nocardia, which obviously is not visible to the naked eye. So as a result of trauma, the organism gains entry through the skin into the human body and can lead to a plethora of cutaneous and subcutaneous manifestations. The characteristic histologic feature of Nocardiosis is an abscess with extensive neutrophil infiltration along with extensive necrosis, surrounded by what? Surrounded by granuloma formation. So initial response to the infection is going to be the neutrophil infiltrate, obviously, and there will be extensive necrosis. Granuloma formation will obviously take some time. It's a chronic event. It will take some time. Okay? So I want you all to remember N. पहले N से नोकार्डियोसिस, दूसरे N से न्यूट्रोफिल इन्फिल्ट्रेशन, एंड तीसरे N से क्या हम याद करेंगे? नेक्रोसिस. Neutrophils are fantastic phagocytic cells, but still they are deceived by Nocardia. So Nocardia is able to survive inside the neutrophils, and for this Nocardia has got a three-pronged approach. Now what is this three-pronged approach? Remember, Nocardia inhibits the acidification of the phagosome when the neutrophils carry out the phagocytosis of Nocardia. Then the second approach is Nocardia inhibits the fusion of phagosome and lysosome, and the third approach is Nocardia neutralizes the oxidants present in the neutrophils. As far as immunity against Nocardia is concerned, remember CMI, that is cell mediated immunity, is very, very important. Very, very important. So what are the risk factors for development of Nocardiosis, that is disease by Nocardia? Anything which jeopardizes or diminishes what CMI is a risk factor for disease development by Nocardia. For instance, somebody who has developed AIDS, that is an HIV infected individual who has developed the terminal stage of the HIV infection, that is acquired immunodeficiency syndrome. Then somebody who is taking steroids, organ transplant recipients, and tuberculosis.